Hello, everybody, and welcome to the 29th Inspiration Exchange session. For those of you who don't know me by now, my name is Fergus Imrie, and I am a postdoc in the Schaar Lab. And together with Tennyson Liu and Mihaela, I organize these Inspiration Exchange sessions. I'm delighted to welcome you today for a session on treatment effect estimation and machine learning for clinical trials. Before I tell you any more about today's session, I also want to tell you about the summer school that the Cambridge Centre for AI in Medicine is running this summer, co-organized by Professor Mihaela van der Schaar and myself. So following the success of last year's summer school, where we had several hundred attendees, this year we will be repeating the summer school again in September. And who should attend? We think the summer school is can be for everybody, and we'll be presenting work on cutting edge machine learning as well as hearing from clinicians about how this is being used in healthcare and how it can be used to transform healthcare. Topics covered, we'll be talking about data-centric AI. We'll talk more than just we're able to cover in today's session about treatment effect estimation. We'll also discuss causal deep learning, automated machine learning. We'll have hands-on sessions where we will, together with participants, get the opportunity to use cutting-edge machine learning methods and much more, such as keynote talks on large language models and much more. If you're interested in finding out more and signing up, I would encourage you to visit the link shown on the slide. Back to today's session. So in the first half of the session, we're going to begin with an introduction by Professor Mihaela van der Schaar, and who is going to discuss where we are on the road to personalized therapeutics. And then we'll hear a couple of presentations from one of the PhD students in our lab, Alicia Curth. In the second half of today's session, we'll have a roundtable discussion with two panelists from industry, who I'll introduce more fully later on in the session. And we'll also have our Q&A session with all of today's speakers. As always, we'll be taking questions for the Q&A in our Slack channel, which will be posted in the chat if you've not had the opportunity to join it yet. So please do post any questions you have for today's session in there, and we will do our best to answer as many as possible, both during the session as well as after the session if you've posted in Slack. So without any further ado, I'd like to pass over to Mihaela as our first speaker.
I'm really delighted to have this session today. We had over the years quite a few sessions on machine learning and what it can do for causal effect inference. And more generally for our lab has been a quite long 8-year-old journey. And I wanted to take a few minutes to just reflect before Alicia starting to talk about research that she has been doing over the last few years, to just reflect on this 8-year-old journey and talk about what's next. Really, this has been an effort that has been ongoing in our lab for many years with many brilliant former students, with some current students, and with the amazing Alicia Curth, who will be talking about later today. So multiple generations of students involved in this area. And the reason why we focus so much on causal effect inference using machine learning is that it's such an important topic in precision medicine. And if we go to how medicine is practiced today, we all know that randomized controlled trials have many limitations. And one of them is the fact that even though current treatment decisions are often based on the average effect of a treatment as observed in a randomized controlled clinical trial, the treatment effects that we observe are often heterogeneous. And there are limitations, hence, to focus on clinical trials alone when describing personalized therapeutics and doing personalized therapeutics for an individual. We know that RCTs often recruit non-representative patients. They are focused on very specific questions, and hence they are unable to capture the complexity of healthcare. They are small sample size, often time-consuming, and some trials cannot be conducted even because they would be unethical. So clearly, we would like to move a patient-centric approach to complement these clinical trials that's based on real-world observational data, whether it's from electronic health records or registries, which is able to capture the complexity of healthcare and is scalable and adaptive and will allow for fast deployment in a cost-effective way. So the type of problems we would like to solve are to estimate the treatment intervention on a particular individual. Now, this is a whole agenda that doesn't include only giving a treatment or not, but also selecting across multiple treatments, determining, for instance, at what dose we should give a particular treatment, and even when to start treating, for how long we should start treating, when should we switch to another treatment or therapy. These are all falling as part of this agenda of causal effect inference. And we know that this problem is hard, and is a problem that not only the machine learning community that this community is part of has looked at, but more than that, the origins of this field are in statistics and econometrics. And you see there one of my favorite books on causal inference. So why should machine learning and machine learners like us even bother attempting to answer this question? And my belief is the reason we should engage with this question is, first, it's an extremely important problem in medicine, maybe one of the most important ones. How to treat, when to treat a particular patient. And machine learning can bring to this agenda flexible and powerful model architectures. And also, I think our community is pretty good at thinking out of the box about the reality of real-world problems. And the area of personalized therapeutics is an especially interesting one for machine learners because of the complexity of questions and the complexity of data and how hard these problems are. So this is an area that I continue to be extremely interested in, and I want to present to you a little bit of a future perspective on it. But before I do that, I want to say that this has been a long journey, and it started by me reading a paper of Uri Shalit, who I saw that is online with us today. So I'm delighted to have Uri here, because it is Uri's paper that convinced me to start working in this field. So we did a lot of work on treatments and treatment effects, first in binary settings. Then in our lab, we started to look at making the problem more realistic by looking at multiple treatments, at continued treatments, treatments over time. And this has been a journey that we are still continuing in because the problem is hard. For instance, by looking at work that has happened initially in biostatistics, we started to adapt solutions to the complicated problem of estimating individualized treatment effects over time and trying to answer questions such as how to treat, when to give treatments, and when to stop treatments. And again, our lab has done a lot of research in this area over the past years, and we are still continuing. More recently, we started to look very hard at how we can bring this causal machine learning lens to enhancing clinical trials. And we have had a session on inspiration exchange not too long ago with Alihan Huyuk, especially discussing that. If you have not seen it, please take a look. And this is an area where causal machine learning can join forces with reinforcement learning, inverse reinforcement learning, few-shot learning, and many other areas in machine learning to solve the challenging problems on clinical trials. And if you want to read more about our papers, we have an entire website dedicated to them. But what I'd like to talk about in the next 3 and final slides is what are the next frontiers in machine learning for personalized therapeutics. One of them is this problem of personalized dosing. So it's not only a question of what treatment to give and for how long, but also at what dose. And we have written a small perspective about this, and this is an area where we have just scratched the surface, and I'd like to entice you, the machine learning community, to look at this. Pharmacologists are very quantitative, and they are very keen to join forces with us, the machine learners, to solve problems. And if you want to see 2 teasers in this area, I invite you to see work that we have done a few years back together with clinicians to determine how we can repurpose dexamethasone for COVID-19 patients in the ICU and what dose we should give this to, and more recently on precision dosing for vancomycin. These are just two of the many research problems we can solve using machine learning in pharmacology. The other issue I want to bring your attention is this issue of time. Which treatment should a patient receive, at what point, when to stop, when to switch treatments? And while some work exists, inclusively from our lab in this area, I think a lot more problems remain. And this is especially the case when we have multiple treatments, but also multiple outcomes and multiple competing risks. Because often we may treat a disease over time for one primary indication, but multiple competing risks may appear as a result of this long-term treatment. These are all problems that require careful modeling and where machine learning, I believe, can contribute. And finally, there is this issue of value of information. What information do I need to acquire to determine what treatment I should be giving and when? And how to look at both predictive and prognostic scores over time to tell me how to intervene correctly on a particular patient given their unique characteristics. Finally, Alicia will talk quite some today about how to combine clinical trials and observational data, enormous opportunities there, as well as determining how to robustly test and validate CATE and ITE models. So, final challenge that I'd like to pose to the community is the fact that we often learn from data sets that contain treatments of patients in different types of countries. And we know that different countries have different incentive systems and also different healthcare systems. So the ability to learn from diverse data sets which contain treatments but in different healthcare ecosystems poses yet new challenges. So this represents a vibrant research agenda and an agenda that I really hope that our community, the machine learning community, can join forces with biostatisticians, pharmacologists, and clinicians to really advance this field of precision medicine.
Thank you so much, everyone, for joining us today to talk about pushing the boundaries of machine learning for heterogeneous treatment effect estimation. My name is Alicia. I'm a third-year PhD student with Mihaela. And most of my work, as Mihaela already discussed, focuses on estimating the effect of a treatment on an individual patient. Basically, I often look at questions that involve something like, will a given treatment work for an individual patient? And we do this by learning from observational data in which we have recorded patient characteristics, treatment statuses, and observed outcomes. There are a lot of these problems everywhere from biostatistics to econometrics to elsewhere. And these problems are vast, but what we kind of argue is that the machine learning literature so far has actually extensively focused on a quite small subset of the problems that are available. Maybe most predominantly, the machine learning literature has considered building new methods for learning heterogeneous effects from static data. So that's the very small subset of the problems for which the machine learning toolbox really has expanded rapidly and now contains everything from meta-learners to causal trees, forest neural networks, and things like Gaussian processes. More recently, some work, as Mihaela just mentioned, has started to look at learning treatment effects from longitudinal or time series data. But this set of work, I would say, has almost exclusively focused on one set of challenges, namely those arising due to the presence of time-dependent confounding. And overall, most of this work has focused on learning from historical data. So where the data collection and treatment assignment is not an active part of the problem, but instead this has already been done. And we're just looking at data that we already have available. What we want to talk about today is about pushing boundaries and having a look at new problems. Today, I'll be presenting 3 papers that we will be presenting at ICML later in summer that kind of try to expand on these problem areas. In the first part of the talk, I'll be looking at static data, where we argue that our toolbox has matured. We have a lot of methods available, so what we really should push is our understanding of the model selection dilemma. How do we actually choose which of the many methods that we have available to use in practice? So this will be the first part of this talk. The second part of the talk looks at longitudinal data. And in that part of the talk, we argue that time brings more challenges than just time-dependent confounding. And in our paper, we study a challenge that we kind of consider orthogonal, which is that of informative sampling. Finally, we consider a different data type. And in the last part of this talk, we consider active data collection, for example, in an adaptive clinical trial. So without further ado, let me start with the first paper that we will be presenting at ICML, which aims at demystification of the model selection dilemma in heterogeneous treatment effect estimation from static data, which is jointly authored with Mihaela. In this paper, we consider a standard static treatment effect estimation setup using potential outcomes, where every individual is characterized by some covariates X that could contain, for example, things like age or gender that don't change over time. And every individual is additionally associated with two potential outcomes. A potential outcome is the outcome associated with a given treatment. And we assume that every individual has a control outcome, which is the outcome that they would attain if they were assigned treatment, and an outcome under treatment, the treated outcome, which is what they would realize if they were assigned treatment. We are interested in estimating CATE, the conditional average treatment effect, sometimes also referred to as the individualized treatment effect, where we'd like to compare outcomes under treatment versus outcomes under control. Basically, what we're interested in is the expected difference of the potential outcome under treatment minus the potential outcome under control, given an individual patient's characteristics. However, the fundamental problem of causal inference is that, unfortunately, we only observe one of the two potential outcomes. So we never have access for any individual to both their outcome under control or under treatment. We can only ever assign them one of the two treatment status in a factual world. That means, unlike normal machine learning problems where we have completely observed labels, the label that we're really interested in, which is this difference between the two potential outcomes, is never observed. And that makes both estimation and model selection pretty hard in this context. Now, as I was saying, the machine learning estimator toolbox has been expanding rapidly recently. And all of this work, everything that I'm presenting today, works under ignorability assumptions, which assumes that there are no hidden confounders. But the CATE estimator toolbox that exists today consists of kind of two lines of work. One considers model-specific strategies, which adapt specific machine learning methods for conditional average treatment effect estimation, for example, causal forests, or CFRNet are implementations of specific methods. Whereas there's also a class of methods I would refer to as model agnostic or meta-learner strategies, which construct something like a recipe for estimating CATE using any arbitrary machine learning method. In this kind of part of the toolbox, there's a lot of different learning strategies. You may have heard of things like an S or a T learner or a DR learner, which can each be implemented using any machine learning method of your choice. In this paper, we focus on the second class of methods and ask the question, given multiple CATE estimates based on different meta-learner strategies and different machine learning methods, how do I pick the best one for use in practice? Basically, we assume that we're given some training data. We have our entire toolbox of all these different meta-learner machine learning methods, combinations, giving us K candidate estimators, and we'd like to answer the question, which one of the K estimators is actually the best for my problem at hand? So basically, we'd like to know which one minimizes the root mean squared error between the true treatment effect and this estimated treatment effect, where the true treatment effect is obviously unknown. So if this was a normal supervised machine learning task, we would just use held out targets to evaluate. So in an ideal world, we'd have validation data, and in that validation data, we'd have observed potential outcomes contrast for each individual, and then we'd just compare our treatment effect estimates to the targets that we observed, and we'd kind of just pick the one that has the lowest error. However, due to the fundamental problem of causal inference, we obviously never have access to this potential outcomes contrast. And this means that we need some alternatives for model selection in this heterogeneous treatment effect estimation context. And some alternatives have been proposed recently, but what we argue in our paper is that there's that real understanding of the advantages and disadvantages of different selection criteria is really lacking so far. So in our paper, we conceptually and empirically investigate the strengths and weaknesses of different selectors and highlight that there's a complex interplay between different selectors, candidate estimators, and the DGPs used for testing them. Before I go into the different selection strategies, let me quickly highlight the two different high-level approaches to K estimation there are among the meta-learners. The first one is to estimate K as a difference between two functions. I will also refer to this as the indirect approach. This is basically any strategy that estimates only the potential outcomes and that then sets the treatment effect estimate equal to their difference. This basically makes use of the fact that here that the K is an expected difference, which you can also write as the difference of expectations between two potential outcomes. This approach basically predicts for every individual outcomes under treatment and outcomes without treatment, and then sets K equal to their difference. A second strategy that's maybe a little bit less popular is to find a surrogate and to target the treatment effect directly. This makes use of the fact that even though the potential outcomes contrast is never really observed, we can construct pseudo outcomes using some nuisance parameters such as the propensity score that have the same expected value when we regress them on X. Basically, we just regress a treatment effect surrogate constructed using some nuisance estimates on X and then get a treatment effect estimate directly. So to summarize, an indirect learner estimates the two outcomes separately and then takes their difference. For example, the popular S and T learner takes such an approach. Whereas a direct learner estimates only the difference between the outcomes, so K directly without the potential outcomes as a byproduct. And an example for that would be an R or DR learner. Now, given that, how do we select between different methods? The first alternative that's often kind of a natural consideration is to use factual or prediction criteria. This basically evaluates indirect estimators, so estimators that do output a prediction of the potential outcome separately by their separate outcome predictions on observed, so factual outcomes and held out data. In that case, you basically just use the validation data to predict the factual outcomes and then compare the observed factual outcomes to the predicted potential outcomes of an indirect learner. This is kind of an intuitive approach and only requires observed data, so it's kind of trustworthy because we're not actually estimating anything else. However, it cannot evaluate all types of estimators because it needs access to a potential outcome prediction, which some of the direct estimators do not do. Also note that this is really not well targeted at the quality of the K estimates. Actually, the K doesn't even appear in this validation criterion. So it would be nice to do something else, some alternative where we actually evaluate the K. A first approach of doing so is to use what we will call plug-in surrogate criteria. Basically, we construct a surrogate for this unobserved K by simply fitting a new plug-in estimator on the held-out data and compare that to the candidate estimators. Basically, this means that we use the validation data and a treatment effect estimator of our choice to provide a plug-in estimate that replaces the unobserved K estimator, unobserved K, and compares that to the candidate. This has as an advantage that it can evaluate all types of estimators. However, it does require a good plugin for the K, and that's kind of the problem that we're trying to solve originally. So we're kind of stuck in the cycle now where we need a good plugin estimator to evaluate our candidate estimators. And maybe a bit more nuanced, we think or we hypothesized before doing the study that this type of selector might really prefer estimators that have a structure that's very similar to the plugin. We will refer to this as congeniality bias. So basically, if your selector, your plugin selector is implemented using a linear regression, it might always prefer candidate estimators that are also implemented using linear regression because it has a similar structure. Finally, we therefore consider pseudo outcome surrogates for model selection. These basically use the holdout data to construct pseudo outcomes that have the same expected value as the unobserved target, using only some nuisance parameter estimates. Basically, in this case, you use the validation data to construct auxiliary machine learning nuisance estimates, and then combine that with the observed data to construct some of the pseudo outcomes. For example, the R learner objective and the DR learner objectives are such pseudo outcomes. Then you use the pseudo outcome to compare that to the Kate estimate, Kate candidate estimators instead of the oracle. This also can evaluate all types of estimators, and compared to the plugin, it at least only requires nuisance parameter estimates and does not require a complete Kate estimate. However, we think that this might similarly prefer estimators with similar structures, for example, those trained on the same pseudo outcomes. So basically that the selector based on the R learner might prefer R learner estimates. And we will test this later in our experiments. So we are very interested in understanding when different estimators work, and we were a bit unsatisfied with existing empirical studies that have mainly focused on finding kind of a global best selector, and by doing so, by considering both relatively opaque data sets and opaque candidate estimator sets. Instead, we are really interested in systematic variation across selectors. In our paper, we discuss some desiderata for better design of benchmarking studies, and then aim to answer 3 questions experimentally. Firstly, we'd like to know when do which selection criteria work better or worse, and whether there are any data characteristics that drive relative performance. We'd like to check whether surrogate selection criteria truly suffer from congeniality biases, and whether we actually lose out if we rely on factual validation criteria instead of the surrogate predictions. In our paper, we design a lot of semi-synthetic experiments that have varying levels of nonlinearities, model misspecification, confounding, and Kate heterogeneity, and implement all different learners and selectors using linear regressions and gradient boosting. We tested this across a lot of different settings that I don't really have the time to go into now, but in short, as a summary of our experimental findings, we find that surrogate criteria sometimes, but not always, outperform factual selection, that pseudo-outcome surrogates generally work better than plugin surrogates, and that the most interesting performance differences occur when there is both confounding and a simple treatment effect. Something that I wanted to show now is the answer to the questions whether surrogate selectors are biased towards some estimators, because I think that's a very interesting question. So one thing that we tested is whether selectors implemented using one learning strategy then also prefer estimators of that same learning strategy. So basically, we considered whether, for example, a plugin criterion implemented using an S-learner actually prefers to select models that are S-learners, and it's quite clear here that this is indeed the case. And similarly, for something we call an extended S-learner, also very much chooses extended S-learners. For the pseudo-outcome criterions, we found this to be a lot less pronounced. So there's a lot less evidence that the pseudo-outcome selectors actually prefer their own learning strategy. Similarly, we did the same test by considering whether selectors prefer, implemented using a specific machine learning method, prefer to choose estimators that are implemented using the same machine learning method. And here we considered for selectors all implemented using linear regressions, whether they prefer to select estimators that are also implemented using linear regressions. And indeed, we found that all the plugin criteria are very biased towards choosing estimators that are implemented using the same method, while the pseudo-outcome criteria display a lot less of such behavior. We then also considered why we lose out when we're using only factuals, but I will defer to the paper for that. Overall, our takeaways are that from this paper, that the Kate model selection problem is a really complex one, and that there's really an intricate interplay between candidate estimators, different selectors, and the data used to compare them. And there's definitely no magic bullet, no selector strategy that always works best. If you're interested, please do check out the paper, because a lot of the benchmarking results are too much to now discuss in detail. But I do think a good takeaway is that there's a lot of room for even more understanding of this very complex problem, and also better solutions. Maybe there is a magic bullet out there and we just haven't found it yet. With that, I'd like to close the chapter of static treatment effect estimation and do a little switch to longitudinal data and our paper on accounting for informative sampling when learning to forecast treatment outcomes over time. This is a paper that was jointly authored with Toon van der Schuure and Wouter Verbeke from the KU Leuven, and will also be presented at ICML. Here, we are now switching to longitudinal data, because often in practice, we actually have longitudinal or time series data available, for example, from electronic health records. And if we have such longitudinal data available, we might actually want to forecast entire trajectories of outcomes under different treatments. Some recent machine learning work has considered estimation over time, including lots of work from our own lab, as Mihaela mentioned earlier. But this set of work has focused solely on effects of time-dependent confounding. What we argue in this paper is that confounding is not actually the only challenge present in such data. Instead, we consider settings where patients are observed irregularly and informatively over time. That means that we observe certain types of patients more often. For example, in practice, we might observe patients that are currently being treated more often because they are being monitored more closely, or we see patients more often when they're sick than when they are not. Therefore, we consider here the challenge of learning under informative sampling. And by doing so, we basically expand the allowed sampling mechanisms that are implicitly considered in recent work. Most commonly, including the work from our own lab, is a setting that considers regular sampling, where observations are kind of equally spaced and regularly observed. But more recently, a paper from our own lab presented actually at last year's ICML, proposing a method called TE-CDE, has considered estimation of continuous time treatment effects under irregular but uninformatively observed sampling. What we consider in our paper is settings where sampling can be informative of some measured attributes. So as I was just saying, it might be that individuals are observed more often if they're currently being treated or if they had worse outcomes in the past. And we explicitly observe for the so-called observation intensity to be non-constant and to vary with histories. However, importantly, we rule out hidden attributes that also influence outcomes. So similarly to the no hidden confounders assumptions, we here make a no hidden information assumption. So why would we care about informative sampling? Basically, what we say in our paper is that informative sampling in the way that we just described it results in covariate shift. And that means that there's potential bias when using empirical risk minimization with observational data, because what we observe is not actually the population we'd like to optimize for in theory. That means basically that for individuals that are more often observed, so individuals that were sampled more often, we make better prediction, whereas our predictions for individuals who are not observed often enough do not get high enough weight. The general solution that we consider in our paper is to use inverse intensity importance weights. Basically, we consider settings where we can estimate the intensity of observation. So where specific individuals, we estimate how likely an individual is to be observed. And if they're very unlikely to be observed, we will give them higher weight. We do this within the TE-CDE framework of a paper from our lab from last year. And we augment the loss function of this neural CDE for treatment effect estimation with inverse intensity weights. In our paper, we have two different implementations for this. One of them is a two-step approach where we simply have a separate intensity predictor network and then use that to weight the loss function of TE-CDE. And we have a second version that's a multitask version where we jointly learn the intensity and the outcome predictions. In our experiments, we find that this kind of inverse intensity weighting indeed greatly improves the performance of TE-CDE when sampling is very informative, which means that it is a good thing to correct for this in practice. So from this paper, we take away that longitudinal data indeed presents many more challenges than just the presence of confounding-induced data shifts, and that different degrees of informative sampling can indeed equally hinder the estimation of personalized effects in practice. If you're interested in more discussion of the inherent assumptions, our methods, and our experiments, please do check out our paper. And in general, we note that learning in such settings requires very strong assumptions on the mechanisms for confounding and sampling to ensure that effects are identified, and similarly, very strong assumptions on any other sources of missingness. So it would be very interesting in future work to relax some of these, to make this more applicable to real world data sets. Now, I hope you're still with me. We now have one more paper to go, and we will now move to this last part of the considered problems, which is to consider different data structures and to consider adaptive identification of populations with treatment benefits in clinical trials. This paper was co-authored by Alihan from our lab and, of course, Mihaela. So in this context, we're still motivated by treatment effect heterogeneity, and now we're considering treatment effect heterogeneity in new treatments, because a new treatment might not work equally well for everyone if there is heterogeneity. Previously, and the rest of the talk, we've kind of considered settings where we have historical data available, but obviously when we have a new treatment, we might not have this kind of historical data available. So instead, maybe we'd like to figure out whether a treatment works actively already during the clinical trial. This is very important in practice because treatment effect heterogeneity can also really pose a challenge to the success of clinical trials, testing the effectiveness of new treatments if only small subgroups benefit. So basically, if the average effect is negative and this masks out that this treatment would have actually worked really well for a good group, but it's in the minority, that means that this group will never get the treatment in practice. And conversely, treatment effect heterogeneity may also mean that the quality of treatment decisions in practice are low when prescribing a drug that has been proven to be effective on average for the whole population, but that there's actually a small minority group for whom the treatment really doesn't work, but that was also masked by the average. Now, as I was saying, much of the recent interest in machine learning on the treatment effect estimation problem has been on discovering effect heterogeneity from logged experimental or observational data. But not so much attention has been given to the active counterpart, so on designing experiments to discover populations that respond well to a treatment. The bias statistics literature, on the other hand, has proposed and extensively studied so-called adaptive enrichment designs for clinical trials, which allow hypotheses and enrollment criteria to change based on interim analyses of the data. However, in such settings, the adaptivity and flexibility is usually limited, and there are few pre-specified interim analysis points and a small number of subgroups considered in these designs. What we wanted to do in this paper is to instead consider whether we can use ideas from the pure exploration machine learning literature to design adaptive trials that are more flexible and efficient. So in our paper, we take a pure exploration perspective on adaptive enrichment designs and consider a problem setting, it's for now a very stylized problem setting where we have K, disjoint, and pre-specified subgroups that have different treatment effects theta. And we're also interested in composite subpopulations that can consist of multiple populations at a time, where the treatment effectiveness is given by the weighted average across the treatment effects within that subgroup. Our objective is to find what we will refer to as a good subpopulation. And a good subpopulation here is a subpopulation where the treatment works, so where the treatment effect is larger than 0. And we'd like to have confidence in this discovery, so we'd like to control type 1 error, but we're also working under a budget restriction. Finally, we do also allow discarding of small effects below a minimally clinically relevant effect size. So we allow, for effects that are very close to 0, we allow them to be discarded. And what we argue in our paper is that this form of problem is very clearly related to some pure exploration bandit problems. So a bandit problem is usually considered in the literature of online experimentation, often in the context of A/B testing of advertising, different ads, for example. But pure exploration bandit problems are slightly different from your standard bandit, in that these are bandits that do not exploit at all. So they're not repeatedly playing the best arm, but they are only there to explore, to find options that are good enough. And in particular, we're here interested in problems that are closest to thresholding bandits and good arm identification. So while our problem is clearly related to pure exploration bandit problems, our setting has some unique characteristics because we're in a clinical trial. In particular, we are subject to constraints on both budget and confidence, whereas these problems usually consider only one of the two. our available budget is usually very limited relative to, for example, online advertising settings, because we're obviously considering real patients and real people. So this really necessitates an early focus on most promising groups. And we often only need to prove effectiveness across a composite subpopulation on average. And finally, we make a real distinction between a null threshold and a minimum relevant effect, because there's a distinction between type 1 and type 2 errors in a clinical trial setting. In our paper, we then adapt ideas from the peer exploration literature to propose two approaches to good subpopulation identification. And in this, we assume a very stylized setting where outcomes are observed immediately and subgroups to enroll can be chosen freely. And basically, without going into too much detail, we consider an algorithm that works group-wise, so identifies good groups one at a time. And then we consider a second algorithm that does subpopulation-wise identification, where we identify good subpopulations jointly by just eliminating populations that don't work well. Each of them have advantages and disadvantages that we test empirically in our paper. But for now, given time limitations, I'm just going to close by saying, due to its unique characteristics, this problem of identifying good populations in clinical trials is actually a really interesting machine learning problem to study in more detail. And there's lots of room for better theoretical understanding of these different algorithms and different strategies. Also, to make some first progress, we've made many really simplifying assumptions here and would be interested in how we can get to more realistic scenarios, including delayed responses and subgroup discovery. If you're interested in more detail, please do check out the paper. There's a lot more in there that I couldn't cover right now, because now I'm just quickly going to close with a couple of concluding thoughts, kind of reiterating that we think that the machine learning literature so far has focused on a very narrow set of problems in the treatment effect estimation context, while in practice there's a much broader set of challenges that arises, and it's time to move our problem settings a little bit closer to reality. And if I could pose a question to the audience here, we are definitely very curious to learn more about other neglected challenges that are there for the application of machine learning in treatment effect estimation problems in your practice. Overall, I'd really like to thank AstraZeneca for the support I've been given during my PhD, during which lots of this work was done. And if you want to learn more generally, please do check out our research pillar on our website on individualized treatment effect estimation. Thank you so much for bearing with me through this talk, and I'm looking forward now to the discussion.
Without any further ado, though, I do want to move now to our panel discussion and introduce our two guests for today's panel discussion, which will be led by Mihaela. The first is Dr. Emre Kiciman, who is a senior principal researcher at Microsoft Research. And we're also joined by Dr. Tom Diethe, who is the head of the Center for Artificial Intelligence within the Biopharmaceuticals Research and Development team at AstraZeneca. And we are delighted to be joined by both of them today. Mihaela, if I hand over to you for the panel discussion.
Yes, thank you so much, Emre and Tom. I'm going to start with you, Tom, with a question. I know you are very interested and have thought a lot about causality in general, not only causal effect inference, but also the general work of causal discovery. How do you think that these two worlds of causality can come together to enable better treatment of patients with all sorts of diseases?
Hi there. So first of all, thank you very much for the invitation. It's great to be here. Yeah, so I think I mean it's clear that that causality is an extremely important question in what we do, and something that I think is kind of often kind of swept under the carpet, or we just sort of say, yes, we know that it exists, and maybe is something we'll deal with later. I think in terms of you know, there are quite a few different areas which where it's really important in for the pharma industry, but I think, you know, running clinical trials is is a clear one, especially when patient safety is concerned as well. We think a lot about is our drug working, but we also have to think a lot about potential adverse events, and whether we're correctly ascribing a causal relationship between, was it the drug or was it something else? And it's often very, very, very complicated because there's so many individual differences. There's only partial knowledge of what's going on. Causal discovery is I don't know, it sometimes feels like an almost impossible task with the levels of complexity that we have. But I think it's been really exciting to see developments in the area. I think particularly applying modern machine learning approaches to it, and I think that started to move the question to something that we might be actually able to start applying some of our methods in reality rather than just in sort of theoretical situations.
Thank you, Tom. Emre, I know that you are working a lot with large language models in recent times. And of course, the world of machine learning has been changed forever with the emergence of these amazing large language models. How do you feel that large language models can play a role, not only on causality, but also on causal effect inference? What are your thoughts on that, and how should we engage with large language models?
Thanks very much, Mihaela, and thank you again for inviting me to be part of this discussion. For large language models, as I was listening to Alicia's talk, I think the types of connections I see there are an ability to use the domain knowledge that's captured or memorized by large language models to help set up the initial set of confounders so that we can have more confidence that before the estimation step and the model selection, that we are setting up the correct causal frame and the causal assumptions correctly, that we're not only relying on the human expert, but also have some way of checking. Another thought that occurred to me while listening to Alicia was whether we might be able to use large language models to help identify a broader set of, say, negative controls, for example, to help with identifying measuring the quality of models. For example, making sure that we're getting a zero treatment effect where we, based on prior knowledge, believe there should be a zero treatment effect.
Really, really interesting. Lots of different ways to think about these large language models, and I think much of our community will be involved with integrating them. So I want to turn back to you and ask a little bit different question. So you in AstraZeneca work in a group that has very different types of researchers involved, not only machine learners, but also statisticians and pharmacologists. How do you feel that this quite diverse group of quantitative researchers could come together? What is a good way for us to collaborate and communicate across communities? And of course, our audience is a machine learning audience. So do you have any food for thought and any advice on how we can engage with collaborators and colleagues interested, for instance, in causal effect inference in other areas? Statistics, biostatistics, or pharmacology, or even beyond?
Yeah, no, that's an interesting question because, sometimes people can feel separated by a common language or something like that. I think, we, for example, in our biostats, make heavy use of Bayesian inference methods, but even there, I have a background in Bayesian inference in a machine learning context, and I find we are, often talking across purposes because, they're very, very heavily in the Monte Carlo world, and you have to, and they sort of maybe just talk about their naming of things is slightly different. You know, what's a parameter and what's a variable and things like that is kind of things that you might think are obvious, not. So I think, you know, sometimes it just takes a little while to make sure, and being like super precise on, you know, any time that you're going to put a formula on a slide, or you know, trying to speak a mathematical language to make sure that everybody is on the same page, because, you know, you sort of just assume, oh, everybody knows this, oh, these people are experts here, and just that translation between the two can be the difference between losing your audience in a meeting very quickly. So I think, you know, we had to work extra hard at that to make sure that we really do have a common language. And I think that sort of, you know, then that sets a good bedrock for anything else, because then people have good things to latch onto. I think, you know, the same thing can be true in causal inference as well. You know, you have different people coming from, you know, econometrics and from machine learning, and coming at it from like different angles, and again, like certain terms that you might assume to be shared terminology are not always the case.
I can also propose an alternative. I would advise everyone to do what I did. Recruit somebody like Alicia, who is an econometrician turned statistician turned machine learners, and then you have the Rosetta Stone across the different fields. So I'd advise everyone to do that. I want to tell Alicia is still 1 year to go, so don't try to get her yet. Emre, as a researcher in Microsoft, who is clearly a company that does a lot of work, especially nowadays, on hardcore AI and hardcore machine learning, but applied to a variety of topics. So not only medicine, but also other areas like advertisement, like Alicia mentioned, and others. Where do you think this causality and causal effect inference methodologies would make the biggest impact going forward, and why?
Yeah, you're right. So we do spend a lot of our time thinking about causality as a horizontal platform, a technology that can, you know, that we're using it for our advertising business. We're using it for, you know, agricultural analyses and precision agriculture and soil carbon modeling. We're using it in health-related problems as well. And I see there is a core set of you know, methods, fundamental methods that span all of these. And then there's an opportunity to build a domain-specific layers atop that help with, for example, bridging some of the language issues that Tom mentioned, not only language, but also common assumptions that are made in different verticals. So there's an opportunity then to specialize, and I think you can reach much closer and get closer to the value for the people who want to use these tools if you are thinking from a domain-specific mindset. I think the places where there's the most immediate opportunity to have an impact with these technologies is in the places where there's already data availability and where the problems are already being framed with a causal perspective or a decision-making perspective. Now, where there's the most immediate opportunity isn't necessarily the place where there's the, you know, biggest kind of long-term opportunity, though. But I think that there are, even in the places where maybe things are a little bit more difficult, like in health, there are clearly milestones we can get to in the short term, so that we can get to the final run. For example, I think it will be a long time before people trust anything but RCTs for, you know, big regulatory approval. But in the meanwhile, there's lots of work to do in, you know, post-marketing, post-approval surveillance, in augmenting RCTs with real-world data in the context of, for example, rare diseases, And also when there's urgent conditions like we saw with vaccine development during COVID, where the urgency required us to lean much more heavily on observational data than maybe we would have otherwise.
Thank you. A final question and a quick answer such that we have time for the audience. So you already answered it a little bit, Emre, but I'll put the question to both of you. What is one problem that you would like to give to the machine learning community to solve in this area of causal inference? And this is a little bit what Alicia's question was to you as well. So can each one of you give us one problem that you think we as a machine learning community should look at? And Emre, maybe I start with you and then I move to you, Tom.
For me, I am most excited right now about this new frontier of using large language models to improve how we think about causality. And the puzzle that's on my mind right now is thinking about how do we think about the gap between how people in natural language terms talk about their causal problems in a domain-specific way, and how to map this to not just the 3 or 4 or 5 different specific well-framed causal tasks that we've defined as academic communities, but really think about the spectrum and what's in between these tasks. The things that we naturally describe these when we ask our natural language question, but then it falls in between and we'll snap it to one problem or the other. And I think that I wonder whether there's an opportunity to think more broadly with kind of the new capabilities we have now.
Very much agreed with you. Sorry, I couldn't stop but jump. I completely agree with you.
I'm going to go for a very kind of specific one to us, just as a counterpoint. So I think one area of great interest at the moment is around combination therapies. And, if you thought causal inference was complicated with just a single treatment effect, then having drug-drug interactions in there, plus all of the things that that Alicia presented early on, I think it is a really, really important and interesting area at the moment.
Very interesting challenge, agreed. Thank you so much, Tom and Emre, for your thoughts and for our discussion. Now, Fergus, I let it to you to get the audience to ask questions to our panelists and to Alicia.
Thanks, Mihaela, and thanks, Tom and Emre, for the discussion. I think we've got a couple of questions in the Slack currently for Alicia that we'll take first. So if anyone else has any questions either for Alicia or for either Tom or Emre, it's not too late to put them in the Slack. But the first question I think comes from Dennis. Dennis, do you want to unmute yourself and ask Alicia your question?
Yes. Hi, everyone. Thank you very much for the great presentation. And yes, I had a question for Alicia regarding the paper on informative sampling. So you used, in your approach, you used this inverse intensity rating method. And I guess this was a bit inspired from this inverse propensity method. So there's this second approach for which you can deal with the confounding bias, learning these unbalanced representation. Did you also consider using this approach, or do you think would it make sense?
So we considered using it, but we indeed didn't do it. That would be one of the next steps to maybe to try if that could work better. Having said that, the multitask architecture is obviously optimized with respect to, so in that multitask setting actually wouldn't work because the multitask architecture is optimized to predict intensities, whereas like a domain adversarial representation actually needs the loss function to be the opposite. But I think it's still something that could be tried to, I'm always a little bit skeptical of using only balancing representations because sometimes that can also get rid of information that you actually need. But I think an interesting question would be to combine intensity weighting with some kind of adversarial objective to make sure that you get rid of any information that you don't really need for prediction. So that would be an interesting next step, but but there were so many moving parts already in what we had that we decided to go with the simplest approach.
Thanks a lot.
Thank you for the question.
Thanks for your question, Dennis. San Mati, I think you also had a question for Alicia.
Yeah, hi. So I also have a question for Alicia regarding inverse probability of treatment weighting. So how sensitive do you think that can be when there are outliers in the data? Like, can outliers have a disproportionate impact on the weighing process, or do you suggest like first removing them and then assigning weights? So in that case, wouldn't that also remove those data points?
Yeah, so what we actually did in practice is we used truncated weights. So if anything was a huge outlier in the sense that it was basically almost never observed, and would therefore, because inverse intensity weights, obviously, if you divide by a very small number, those weights blow up. We actually truncated those weights and set them equal to a constant. So we kind of took care of it that way. We do have some experiments in our paper where we actually, so once you make something very, very informative, so in our data generating process where we had semi-synthetic data, we had a parameter that's where we kind of controlled the informativeness. Once there's so much information that basically there are certain groups that are almost never observed anymore, then you see that such a weighting approach indeed deteriorates because there's simply not enough information in what is being observed. So this is definitely this is in some ways definitely sensitive to very, very strongly informative sampling. So in that case, one might want to do something else. I don't know if Dennis's suggestion from previously, maybe some kind of balancing representations would actually work better in that case, because at least you're not blowing up your loss as much. But that would definitely be an interesting question to further investigate.
Yeah, thank you.
Thank you so much.
Thanks, Sanati. I don't know, I think that's all of the questions we covered on Slack. I don't know if anybody in the audience or possibly even Alicia, if you have any questions potentially for either of our panelists or vice versa. If you don't think we've got time for probably one more question, if anyone has anything burning they'd like to ask.
Well, if there is nobody, I have a burning question for Tom. I want to ask Tom what he thinks about these ideas of sampling in clinical trials, especially. So currently, as far as I understand, in clinical trials, patients are followed in a particular pre-specified way. And of course, this is on one hand good. On the other hand, it puts an enormous burden for already very sick people. So the question is, do you see opportunities to think differently about this problem of monitoring and monitoring over time in clinical trials? And are there ways of thinking? And is this a good machine learning problem?
Yeah, I mean, I was looking at that and I was thinking, you know, this is so tricky because of the highly regulated nature of it. And, you know, changing major things about trial protocols in that setting, you're basically going to encounter a huge amount of resistance. So I was kind of looking at that, and I completely see why you would do it. But yes, I think there is a bit of an uphill battle. I think there might be, you know, there might be some cases where you have a bit more freedom than others. So, you know, the majority of trials will be, the thing is, like, you also have to think for the majority of trials, everything is so constrained. You know, they're really, really trying to keep down any complexity in the trial adds time, adds costs, and makes everything. So like when every single trial is trying to be like squeezed and in every direction, they want as few people involved, they want as few, you know, the documentation to be as fast as possible, everything about it to be as simple as possible, then, you know, for the majority of trials, it's going to be very, very hard. But there might be some where, you know, maybe the endpoint is more exploratory. Plus, also, you know, you maybe have more of a chance in a phase 2 where, you know, it's less of a big deal. They're much smaller trials. That you might be able to get something going. But yes, I mean, honestly speaking, that is going to be a hard challenge. I mean, there's so many hard challenges around anything that's kind of labeled as precision medicine, and I guess like this is it, you know, it's not what people normally think of it, but sort of like adaptive trial design or something like that is extremely challenging. Yeah, I don't know. I know I don't know an instant
Thank you.
answer, but yeah.
I think that just about brings us to the close of today's session. And before we go, I'd like to say a massive thank you to all of our speakers today, including Mihaela, Alicia, and our 2 guests, Emre and Tom, for their presentations and for joining us for discussions. I think that's definitely a lot of food for thought for myself from today's session, and I hope for everyone in the audience as well. Today's session will be on YouTube relatively soon, but unfortunately we are going to be taking a break for summer soon, but we'll be back for the 30th Inspiration Exchange session in September after the summer break. And so please do join us for that when we return. And thank you everyone once again for joining us, and enjoy the summer. Thank you.